All right, thank you.
Okay, thank you, Jeff.
Yeah, I'm a recent postdoc in Sundar's lab, temporarily. I'll be talking about most of my PhD work here. So I've been working on the root-associated microbiome of rice. And so what do I mean by a microbiome? So it's a collection of microbes and the habitat that they colonize. This is mainly used for the bacterial and archaeal portion of the microbes. This is widely popularized through the Human Microbiome Project. So I guess it's been known for a while that microbes in your gut can help you metabolize your food, synthesize vitamins, but we'll be talking about plants today. So plants also have microbiomes. So here's a microscope image of a rice root. You can see the root cells here. Each one of these green dots is a specific or a bacteria that's been probed with a eubacterial probe. So these microbes are important to plants. Of course, we know about pathogenic microbes which can hurt the plants. We know about a few beneficial microbes that can help the plant to acquire nutrients from the soil, to be protected from these pathogens, and to promote general growth. There's also these microbes which have no effect whatsoever on the plant, but may affect these other microbes that have important functions. So this is a very complex network which we're trying to untangle, and how all of these guys work together on the plant. And we've been doing this in rice. So rice is a very important agricultural crop worldwide. It's actually grown in a few areas in the United States, in the Mississippi Delta, in Arkansas, Louisiana, Texas, but also, fortunately for us, in Northern California, making all my field experiments easier. It has a unique growth habit in that it's semi-submerged underwater. This is unique for large crops such as rice. So when we're looking to see which microbes live in and around the rice roots, we take a culture-independent approach by sequencing the 16S ribosomal RNA gene. So basically the way this works is you take your sample, whatever it may be, whether it's the soil or the root, you extract the DNA, and then we can amplify the 16S ribosomal RNA gene with PCR. We can do some high throughput sequencing, and basically the sequences we get back, we can identify which microbes are there. And so we basically get a catalog of tens of thousands of different types of microbes that are in our samples. So to give a general outline of what I'd like to talk about today, I'm going to be talking about just some basic, just give you a little review on the basic factors that we found to be important in the root-associated microbiome of rice. And then I'm going to talk about how rice compares to other plant species. And then I'm going to talk a little bit about the dynamics of these microbial communities. So we start out by asking some very basic questions of what do microbial communities look like out in the soil, just outside of the root, so in the rhizosphere, versus these microbes on the root surface, which we call the rhizoplane, and versus those communities that are on the inside of the root or the endosphere. So how do all of these communities compare to what's just out in the soil? Also, let's say you're growing in a field in Davis and a field in Sacramento. Do these different fields have different microbes that associate with the roots? So how does geography affect the microbiome? And also, if you're growing different genotypes of rice, how do those different genotypes vary in terms of their root microbiota? So I'd just like to summarize what we know about this so far. So let's say you're growing in Davis and you're growing in Sacramento. The two soils there will have two completely different microbiotas. But then what the plant assimilates from those soils in terms of the microbial communities are just subsets of these microbes. So if you're looking at a cross-section of the root here, let's say around the outside in the rhizosphere, it'll be very similar to the soil. But then on the inside, it's a much reduced community, a much less diverse community but still derived from the soil. And we see that these differences between the soil types are reflected in those communities. However, if you're looking across these different genotypes, we see only a small level of variation due to genotype. So even though there's plenty of phenotypic variation above ground, we only see a very limited set of variation in the root microbiota. So this led us to ask this question: if we're looking outside of rice and at interspecies variation, do we see more variation in the rice in the root-associated microbiota of plants? And so fortunately for us, there's rice is not the only thing growing in a rice field. There's also plenty of weeds growing out there. So what we did was we went out to the field and we collected a bunch of different, or three other plant species. So we collected rice, we collected a sedge, Heteranthera, and then a dicot Bacopa, and we compared all their microbiomes to each other. And so we knew that there's very little variation between different genotypes of rice, but how does the variation look if we start stepping outside of rhiza? So here's the results here. So this is a principal coordinate analysis, very similar to PCA. And basically, the way you read this is one sample represents one microbiome that we sampled. And if, let's say, one sample is close to another sample, it means that on this graph, it means they have very similar microbial communities. But if, let's say, this point and this point, they have very different microbial communities. So we get several sources of variation coming out here. One is that you can see that all the rhizosphere samples are over here. So that's the soil that's just outside of the root. All the endosphere samples are over here. So these are the microbial communities on the inside of the root. And so all these different colors represent the different species that we're looking at. And so we can see in the rhizosphere and the endosphere that rice is an outlier compared to these other plant species that we're looking at. So just to sort of summarize this very quickly. So now what we notice is that the variation between rice genotypes is very small, but once you start looking at other species, the variation becomes much larger. And I sort of skipped over this, but we think this may have functional implications. We notice that the rice plants have much more methanogenic archaea. So these are the microbes that actually make methane than these other plant species. So this may be functionally relevant. And we also noticed there was no correlation between the phylogenetic distance between these different plant species we looked at and the microbial communities that they assembled from the soil. So for instance, this Bacopa, which is a dicot, was the closest to rice that we found. Something else that we noticed from this data, and maybe you saw it too, was that these pink dots represent samples from soil where no plants were growing. And so this came from a rice field. So these soil samples have a nearly identical microbiome to the rice rhizosphere. And so we put forward two hypotheses to explain this. One is that maybe through the process of domestication, rice has lost its ability to differentiate its rhizosphere microbiome from the bulk soil. That is soil that doesn't have any plants growing in it. Or another hypothesis that we came up with is if you've been out to a rice field, you notice that it's very densely planted. So maybe if you're growing over multiple seasons, all the roots grow together, and technically everything in that field becomes just like the rice rhizosphere. So we're following up on both these models. I'm going to talk a little bit about this one today. So I call this the domestication model, the soil domestication model. So we wanted to investigate this further. So to test this hypothesis, we went out and we collected different soils across California. So we collected what I'm calling domesticated soils. So these are soils from rice fields that have been growing rice for upwards of 8 years. And we collected soils from places adjacent to rice fields, but had not been growing any rice. And we took these soils back to our greenhouse and grew plants in them and looked at the microbiomes that they assembled. And so what we noticed is that the rhizosphere effect is much higher in these wild soils. So rhizosphere effect, and I mean that the difference between the bulk soil communities and the rhizosphere is much higher in these. So you can think of that along this axis here. It's just the difference between bulk soil and the rhizosphere communities that they assemble. So it's much higher in these wild soils than these domesticated soils. So sort of a cartoon depiction of this is if you're looking at a wild soil here and the plants growing in that, those microbial communities on the rhizosphere are much different than these communities out in the bulk soil. But if you're looking at domesticated soils, you can see that these communities are very similar to each other. So this is indicating that these plants in the growing in the field have changed their field's microbiome. So here's another model. I wanted to just give snapshots of this data, so I didn't talk about some of this other stuff. But what we noticed is that between these wild soils, there was much more variation than between these domesticated soils. So these domesticated soil microbiomes had very similar microbiomes, and these wild soils had very different microbiomes from each other. So the model that we're putting forward, which still needs to be tested, is that maybe over multiple years of continuous cultivation, you can sort of take a soil from being wild to having a domesticated state. So there's these other questions we're trying to address. We're going forward with this is how long does this take? If you start putting other crops on this rotation, does that affect this process? And does this happen with all crops, or is it just rice? And of course, functionality. Is this process even important to the rice plant's health? So just to summarize this portion, so plants have distinct microbiomes that are spatially distributed in these different root compartments. The largest driver of these differences is, of course, the various soil types, and this can be affected by how you cultivation practices, such as being organic or conventional. Within species, genotype variation has a very small effect on the root-associated microbiomes. But once you start looking at microbiomes outside of that species, you can find larger differences. And continuous cultivation may actually domesticate the microbiome of that field. Okay. So these have all been what you call a cross-sectional study, or just a snapshot of these different factors of variation that we can explain with the data. But now I want to start talking more about how the microbial communities change over time over the life cycle of a rice plant. So what we did was we just went out to a single field in California, and we collected rice plants weekly and sampled their microbiomes and crunched all the data. And so we hit all of these major developmental points. So germination, this is a really important point of when this is panicle initiation, when the rice plants decide to actually go into the reproductive state. And then we went all the way through senescence. Okay, so this is just an overview of the data. Again, we notice that the microbiomes are distributed spatially. So we get our rhizosphere samples over here. Those are the microbial communities out in the soil just next to the root surface communities, and then the root interior communities over here in blue. So those all separate along the first axis. Along the second axis, we see that rice age is a major factor contributing to variation as well. And so we noticed a trend here, which I'm going to show a little more clearly, in that it looks like these communities are stabilizing at the onset of reproduction, or that panicle initiation stage. So what we did was we fit a linear model through the vegetative portion of the life cycle and through the reproductive portion of the life cycle. So what we notice is that if you're fitting a linear model across that second axis in the vegetative portion, it's changing quite rapidly. So this is a slope estimate. However, if you fit a linear model in the reproductive stage, you notice that it's not changing at all. So this indicates to us that at least 56 to 63 days after germination that these microbiomes begin to stabilize. And so this is sort of, we can depict that with these different phyla distributions here. So we can see that delta proteobacteria, which goes up over the course of the season, and beta proteobacteria goes down over the course of the season. So here's sort of a cartoon model here. So we see that at the beginning of the season and at the end of the season, we get a completely different microbiome associating with the plants, but this changes throughout the vegetative portion, and then it stabilizes panicle initiation when the plant decides to go into reproduction. But still, this is not direct evidence that development has anything to do with the microbiome. It's just a correlation with plant age. So we went out and we planted these different rice varieties that progress at different rates through their life cycle, or develop at different rates. So we have the actual age of the plants here, and then this is the developmental progression. So we have a very quickly progressing variety, Kitake here, a very slowly or a relatively slowly developing variety, Nippon bar. And we collected the microbiomes from these over time to see if developmental stage actually had an important effect. And so here I'm just going to summarize the data. What we notice is if you're just looking out in the soil, there's basically no change over the course of the season. If you're looking in the endosphere, you can see a large change over the course of the season. So these are young plants down here, older plants down here. And if we fit a linear model again across the second axis and look to see how these different genotypes vary, we can see that Kitake actually moves along the second axis much faster than Nippon bar, which is the later developing variety. So sort of the take-home message here is that the root microbiome shifts significantly over the growing season. We notice a stabilization around 8 weeks after germination. We also see that there's this different developing, or plants that develop at different rates actually have a faster progressing or slower progressing microbiome. But this effect is much smaller than just the age alone. So plant age is much more important than developmental stage for determining the microbiome. So with that, I'd like to thank you for listening. I'd like to thank the lab that I work in, so the Sundar lab, the Eisen lab, dissertation committee, Shavana, Jasara in the hallway, all the people that have helped me so far, especially the farmers and all the funding. And thank you.
Any questions? Okay. Let's start from here. I just have a question about, do you know if the farmers are adding fertilizer to their fields and how often, and whether or not that can also affect the community there?
Yes, so we know, so these farmers, so this progression I just showed was only out in one field. And we also have done the same study over multiple seasons and in Arkansas as well. And they fertilize at different rates at different times, and we get basically the same result. But yes, we do know if you're using conventional cultivation or organic cultivation, you're going to get a completely different microbiome. So fertilization types and rates do affect it. Yeah.
Is this on? Yeah, okay. I think I might have had a very similar question I didn't hear. So to what extent domestication of the soil, of the microbes, has to do with agronomic practice? Is that what she asked as well?
Yes, so
flooding and nutrient application and just compaction soil types that are selected for crops?
So that definitely is part of it, but it's also the rice plant itself has a unique microbiome that is coming through in these domesticated soils. And I have a poster back there which I could explain it more thoroughly with, because I use the comparison of these other. Maybe I'll go back and then talk a little bit. So using this study, I could find microbes that are specific to rice. So even though these are all growing in the same flooded environment, there's these rice-specific microbes, and these microbes are coming through very much in the domesticated soils. So it's not just a product of flooding or adding nutrients.
I think I know the answer, but have you looked at any weedy rice and microbiomes of weedy rice?
No, not yet. Yeah, I would really like to look at pre-domesticated, or so Oryza rufipogon and the weedy rice. Yeah.
When you looked at the rice, I'm assuming you looked at the growth conditions of the rice while it was in the domesticated soil versus the wild soil. Any difference in the growth of the plant itself?
So that's a tricky comparison, because these soils do have different levels of nitrogen, or all the nutrients are different in structure. So what we're doing now is we're actually inoculating a common substrate that has all the same properties, nutrient properties, with just small amounts of these microbiomes, to see if those give different quantitative phenotypes we can measure on the rice plants. Yeah.
So to the uninitiated, do they grow a cover crop after they harvest the rice? And do you know, do you look at that at all?
Not in California. So in California, typically what they do is they either flood the field, or what they used to do is they just burn the field. But through climate regulations, what they do now is they typically flood the field, till under the tillage, and then start a new crop. In other parts of the world, they just continuously grow rice. So they can get three or four crops a year.
Thank you.
Thanks.
All right, we've had some wonderful talks this morning.